Aubrey, I think, was thinking when he invited me here, that we might use these zinc finger recombinases that I'll tell you about in the next part of the talk to fight aging. I have to mention to Aubrey that he was not the first one to think of using recombinases as the source of the fountain of youth, but as my graduate student pointed out, it actually appears in an Eddie Murphy movie, so I think it must be a rather good idea. Currently, there are two strategies for genome modification, and many of you will know that one of these strategies was recently awarded a Nobel Prize that involves homologous recombination, which is affected by directing a double-stranded break to facilitate that. And there are two outcomes, either non-homologous end joining, which results in an error typically in that sequence, or homologous recombination that allows you to insert a desired sequence. Now, our interest is to use a rather more directed approach, which is site-specific recombination. Recombinases in nature allow you to do integration, so you can do gene delivery, but you can also do other sorts of interesting rearrangements in the genome, so you can turn genes around, you can move them from one place in the genome to another. So you might imagine that these might be tools that allow us to really rewrite the genome in a very specific way if we can learn how to control their chemistry. Now, there are a number of limitations to all of these approaches. Homologous recombination relies on endogenous repair proteins. It's versatile, but it's rather inefficient. You have to modify the frequency of homologous recombination is in the part per million range. There are now approaches based on zinc fingers using nucleases that facilitate this strategy. Those are zinc finger nucleases. However, that approach suffers from that side reaction, which is non-homologous end joining, which also results in damage to the genome in places elsewhere where that nuclease breaks. Site-specific recombination is controlled by that recombinase protein itself. It's typically rather efficient. Many of you are familiar with Krelok's animals. However, these proteins have a fixed DNA binding sequence that they act on. And so our goal is now to be able to engineer those proteins really to allow the scientists to create a simple machine that will, protein-based machine that will allow you to deliver genes or manipulate genomes in a very specific fashion. Given the time constraints, I won't go over the various other technologies that are out there for delivering genes. However, many of them have significant problems, either in terms of immunogenicity or in terms of specificity or efficacy. So these are our sort of the types of thresholds that we'll need to overcome to develop this as a versatile class of proteins to solve this problem. To address this challenge, again, we've turned to a natural class of recombinases, not the type of tyrosine recombinases most of you will be familiar with in your studies with Krelok's. Those types of recombinases have a less modular nature than those that you see here on the left-hand side of the slide. And they're less modular primarily in terms of their intertwining of catalytic activities with DNA binding activities. So they're, from an engineering perspective, they're very difficult to engineer in a very rational way. However, this other class of recombinases, which is not well exploited in molecular biology, which are the serine recombinases, have a rather modular structure. They are homodimers, just like tyrosine recombinases, but they have this head domain wherein lies most of the catalytic activity of the protein. And they have a separate appendage, which is the DNA binding domain, which imparts a lot of the DNA binding specificity, but not all of the specificity to this protein. So with that in mind, and with this modular separation, let me show you how these proteins work. And this is a movie that was constructed by the Stites lab, who's done a lot of the structural biology on this class of protein. What you'll see at the top, it's hard to see in this projection, there's actually double-stranded DNA here, double-stranded DNA here, and what you see here are two homodimers that have bound to DNA and are interacting via that head region. And what they'll do is they'll actually recombine those regions, if I can get that moving to play. So the recombinase binds to DNA, you can barely see it here, breaks the DNA and makes a covalent bond to it, and then there's a translocation about that head region. So you actually see 4 proteins bound to DNA in this view. So this is the type of molecular machine that we're building using this approach. To do so, we've taken this modular approach that we took also with zinc fingers. We truncated the natural DNA binding domains from these serine recombinases and placed onto them our designer zinc finger proteins, now of course that we can direct to bind any given DNA sequence. These recombinases then, in this view, form a homodimer, but we could make them form a heterodimer as well. So the DNA binding activities of these proteins are directed by the zinc finger binding domains that we can append onto them. There's also this core sequence, this 20 base pair core, which involves some interactions from the catalytic domain that to date are not well described and that we're just beginning now to learn how to manipulate in a rational fashion. However, we wanted to address the question if we could actually use this as a tool to insert genes into the human genome. And we wanted to take an advantage of 2 levels of specificity here. Specificity provided by our zinc finger proteins. That's the sequence that we can directly assemble a protein now to bind. Take that together with the endogenous sequence that the core of the recombinase recognizes, that 20 base pair sequence, and ask if we were to assemble this protein, could it indeed act in higher organisms and deliver a gene in a very selective fashion. You should know that there are more than 50 serine recombinases known, all that differ in that core sequence. And so in this next experiment, what we're doing is we've introduced a site into a human cell line that contains this 20 base pair core of either TN3 or GIN, and then we've appended on zinc fingers onto the enzymatic domain of those 2 serine recombinases. This schematic here shows you the details of the experiment. So we basically have 2 plasmids. This plasmid is expressing the zinc finger recombinase, and that is shown here as a homodimer. This is the plasmid that it will act on. This is the recombination site. That DNA sequence you see here is illustrated here as this element on this plasmid. The plasmid has a pyrimycin resistance gene on it so that we can track the cells that have successfully integrated the DNA. So within the genome of a human cell line, we've placed a GFP protein also bearing a copy of that same site that appears on this plasmid. Now, if this is to work, these proteins should form that same sort of bridge structure, bridging now the incoming donor DNA with the DNA on the chromosome of the human DNA, making that tetrad of sequence. The protein should then bind to the plasmid and the genomic DNA, make a new phosphoester linkage with that, and exchange those 2 strands, eventually then inserting that plasmid DNA at the site of green fluorescent protein. In this case, we designed the experiment so the plasmid can actually insert in either direction. We now have means of controlling the directionality of integration as well. And then the outcome ideally should be either, since we have a promoter element here flanking this recombination site, should be either enhanced GFP, so when that promoter is in line with the GFP, which finds itself in the genome, or diminished GFP when there's interference with that promoter. Now this is what we see when we deliver these plasmids into human cells bearing EGFP. We transfect and select with pyrimycin. This is the original level. This is the cell line expressing GFP at a specific position in the genome. We now add the zinc finger recombinase. It inserts the plasmid actually in both directions. And you see by the FACS profile that you see both a brighter and a more dim population of cells as a result of now this protein inserting that plasmid in a very specific way.
3 more minutes.
We can isolate the I don't have a 30-minute talk, I have a 15-minute
You
talk.
do.
Okay, we can insert the